Okay, so welcome back to the classification of amino acids, and here the classification is based upon the nutritional requirement. Okay, the first grouping, it is essential amino acids or indispensable amino acids. These amino acids cannot be synthesized by the body, and hence it has to be supplied through the diet, and they are required for the proper growth and maintenance of the individual. Okay, so that is why it is known as essential amino acids. Indispensable, it is unavoidable, it is needed for the maintenance and proper growth of the individual, but it cannot be synthesized. Okay, so these are the amino acids which are supposed to be supplied through the along with the food. Okay, so it is arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. Okay, the next grouping is non-essential or dispensable amino acids. We can uh, the body can synthesize on its own, and hence it needn't be supplied through the diet. Okay, so it includes glycine, alanine, serine, cysteine, aspartate, that is aspartic acid, asparagine, glutamic acid, glutamine, tyrosine, and proline. Now that's yet another group which is known as semi-essential amino acids. What happens is um, the arginine and histidine, these are the amino acids which can be synthesized in the body of an adult, but in younger individuals, what happens is these amino acids have to be supplied through the diet. Okay, that is why it is known as semi-essential amino acids. So based upon the nutritional requirement, the amino acids can be classified into essential, non-essential, and semi-essential amino acids. And you can see that most of the amino acids almost like 50% of the amino acids are non-essential, and the other essential amino acid. Okay, now the next classification is based upon their metabolic fate. That is what happens to the amino acid during metabolic pathway. Okay, so we can see that the amino acids can serve as a precursor for the synthesis of glucose. That is, amino acid can undergo metabolic pathway to give to result in glucose. Okay, and there are certain amino acids which can give rise to fat, or there are certain other amino acids which can give rise to both. Okay, those which can synthesize glucose, they are known as glycogenic. Okay, and those which can be precursor for the synthesis of fat, we call it as a ketogenic, right? Glycogenic amino acids serve as precursors for the formation of glucose or glycogen, and they are alanine, aspartate, glycine, methionine, etc. Ketogenic amino acids, on the other hand, they serve as precursors for the synthesis of fats. Okay, there are two amino acids which are exclusively ketogenic. These are leucine and lysine. And glycogenic and ketogenic amino acids, there are four amino acids which are both can be precursors for the synthesis of both glucose as well as fat in the same way. They are isoleucine, phenylalanine, tryptophan, and tyrosine. So this is about the classification. So we saw the classification based on the polarity of the R group, the structure of the R group, then the nutritional requirement of, and then the metabolic fate of the amino acids. Okay, now we pass on to the physical properties. Can see these are the few physical properties of amino acids. Most of the amino acids, they are usually soluble in water and insoluble in organic solvents. Uh, Most of them can form hydrogen bonding. That is, most of them are polar molecules. That is what it indicates. Okay, then amino acids generally melt at a very high temperatures, almost like around 200 degrees centigrade. It melts. Okay, so it is very high. Then optical properties, all the amino acids except glycine, they are optically active. They have optical isomers, and it is due to the presence of the alpha carbon atom, which is asymmetric in nature or chiral in nature. Okay, amino acids, they are both acidic and basic because it contains amino group and as well as a carboxyl group, and it contains a positive and a negative pole, right? So such a situation is referred as ampholytes. Okay, that is both it can accept a proton as well as it can donate a proton. Such condition, it is known as a ampholyte. Okay, so when we discuss about the ampholyte, the reason it is because of the dipolar nature. Okay, it is otherwise known as zwitterion. So each amino acid, they have a characteristic pH, that is specific pH, at which the amino acid, it do have uh, a positive and a negative charge. Okay, so it will be almost neutral, and this particular pH, it is known as isoelectric pH. Okay, it is known as isoelectric pH. So isoelectric pH is a pH at which the amino acid carries or both positive and negative charge. and it means isoelectric pH is the pH at which amino acid like exist in zwitterionic form. Okay, so each amino acid usually has a very specific characteristic isoelectric pH, and isoelectric pH is usually referred as pi. Okay, so this you can see, right? So this is amino acid. What happens when there is a yeah, you can go this way, it becomes more acidic. It is in more acidic condition, means it can the external medium contains more hydrogen ions. So what happens? NH2 becomes NH3 plus. Okay, so at low pH or acidic pH, it is actually positively charged, right? So it is not zwitterionic here. But when further down, when it is the more acidic, what happens? You can see here, right? So this is what happens. Okay, so similarly, higher pH, it results in like the anionic form, and lower pH, it results in cationic form. But at a new pH around isoelectric pH of that particular amino acid, it forms a dipolar one. Okay, so that is one the physical property. Okay, zwitterionic form, it exists at when the pH of the medium is at the isoelectric pH of the amino acid, right? Okay, so the chemical properties. When we speak about the chemical properties, we can divide it into two, that is reactions undergone by the amino acids due to its carboxyl group, okay, and reactions due to its amino group. Okay, when we speak about the reactions due to carboxyl group, the first one is it can form salts, sodium salts for example, with bases, and esters with alcohols. Okay, I hope it is clear, right? Then it can undergo decarboxylation reaction, and when it undergoes decarboxylation reaction, it produces the respective amines. Okay, so you can imagine the decarboxylation reaction of amino acids. It can give rise to various kinds of biologically active molecules. For example, like the just 1 minute, 1 minute. So decarboxylation, that is, it can undergo decarboxylation in the sense it releases carbon dioxide. Okay, so when it releases carbon dioxide, it produces corresponding amine. Okay, and this particular reaction, it is significant in living cells because it can result in the formation of biologically important amines. For example, histamine. Histamine is produced, as the name indicates, histamine. It is produced from histidine by decarboxylation process. Similarly, what you call glutamic acid or glutamate, it give rise to GABA, or what you call gamma amino butyric acid. Okay, that is a neurotransmitter molecule which you may be learning in your physiology classes. Okay, so histamine, again in your immunology classes you will be learning histamine. So you can see all these molecules, histamine, GABA, all are produced by the decarboxylation of amino acids. Okay, now next we have is the reaction with ammonia. So it is actually the carboxyl group of the dicarboxylic amino acids reacts with ammonia to form the amide. Okay, aspartic acid, it is a dicarboxylic monoamino amino acid, isn't it? So there what happens, it is the carboxyl group of this one, it reacts with ammonia to give rise to the amide. Okay, it is asparagine. Likewise, glutamic acid, it gives rise to glutamine, right? So these are the few reactions which is taken up due to the presence of carboxylic acid group, COH group. Okay, so one is decarboxylation, and the other is reaction with ammonia, especially with respect to the dicarboxylic monoamino acids. Okay, now what are the reactions due to the amino group, due to the presence of amino group in the amino acids? The first one, the amino group behave as bases and combine with acids to form salts. Okay, it contains this compound, right? Now, reaction with ninhydrin. Actually, ninhydrin reaction is used for the determination of amino acids and proteins. In a sample, the quantitative determination of amino acids is done with the help of ninhydrin reaction. Now, in the ninhydrin reaction, it is actually alpha amino acid. Okay, it's not D amino acid. Alpha amino acid. It reacts with ninhydrin, actually ninhydrin reagent, and it reacts with ninhydrin to form a purple, blue, or pink color complex, and this is known as romance purple. And amino acid with ninhydrin, it gives rise to keto acids. Okay, and hydrin dantan, and this one it can give rise to the purple coloration. Okay, further reaction, it can give rise to purple coloration. So very important reaction, ninhydrin reaction. It is because of the amino group present in the amino acid. Now the next one is transamination. Transamination and oxidative deamination, it is a metabolic like a process taken up by amino acids. Transamination, it is, as the name indicates, transfer of amino group. Okay, so where does it transfer? Transfer of amino group from one amino acid to a keto acid. Okay, how is keto acid formed? Keto acid is formed when amino group is lost. Okay, loss of amino group, it results in like keto acid, and transfer of an amino group from an amino acid to a keto acid give rise to a new amino acid. Okay, and the donor amino acid becomes a keto acid. So it is a transamination process. It is a very important reaction in amino acid metabolism, right? And the second one is oxidative deamination. So here what happens? The amino acids undergo oxidative deamination to liberate free ammonia. It is again deamination. So there is no an amino acid loses ammonia there. Okay, it is, but the only one thing is it is oxidative process. Okay, so these are the properties of amino acids. So we saw the classification complete, physical properties of amino acids and chemical properties of amino acids in this presentation. Okay, fine. Thank you.